this model here is our old meiosis model. I don't know if you saw it in AMP1 or not. It's like, this model came out like in 1852 or something. I don't know, it's old as hell, but anyway. Here's the model. So meiosis, just a couple of things. Remember, there's 2 rounds of cell division with meiosis. On this particular model, these things that are shown in red and purple here, those are the actual chromosomes, or you can see them a little bit better. Here are the chromosomes shown there. I think everybody kind of knew that. The chromatids, does anyone remember what a chromatid is? It's like half, remember chromosomes can sometimes chromosomes look like the letter X, sometimes they look like the letter I. When they look like the letter X, half of the chromosome is a chromatid. So if you look down here, all right. So this whole thing would be one chromosome. This portion of it would be a chromatid, and then this portion over here would be a chromatid. The part in the middle that holds them apart, holds them together, that's the centromere. Okay. What else can I tell you? Some other things that hopefully you remember from AMP1. What's this structure called here?
Spindle.
That's the spindle, or you can just call it the spindle or the meiotic spindle, whatever you want to call it. What were these little yellow things? Those are the centrioles. The spindle extends between where the centrioles are at the end. Let me go through then some of the actual phases then. So this up here, because you're not even actually seeing the chromosomes yet, this would be interphase. So the cell's not even undergoing division yet at that point. Over here, you're starting to see these worm-like things. These are going to be the chromosomes, right? When a cell is just chilling out, most of the cell's lifespan is spent in interphase. And remember, the chromosomes are like unraveled during interphase. So you can't actually see the individual chromosomes, and that's why it looks kind of grayish here. But when meiosis begins, the chromosomes start to condense. So during prophase, which is what this slide is, or this model, you start to see some of the individual chromosomes, which look like little worms here. Okay. So this would be prophase 1. As you move along to this spot, this is also prophase 1. The only difference here is that the pairs of chromosomes are starting to find each other. Okay? So the member that comes from your mom and the member of the pair that comes from your dad, they hook up, they find each other, and then they do this thing called crossing over. Do you remember that from AMP1, hopefully? This is still prophase, okay? Again, one of these chromosomes would be from your mom, the other one would be from your dad. They find each other, they start sort of aligning themselves together. Again, it's still prophase 1. Over here, once again, we're still in prophase 1, but here you see the actual crossing over taking place. The chromosome pairs, they swap their DNA. They basically flip their arms across and they exchange their DNA with each other. And that's one of the reasons why we have so much diversity among people, why you don't look like your sister or your brother, thank God, right? Because of things like this that lead to a lot of genetic diversity. So that's the crossing over. This is, again, still prophase 1. So if you're keeping track, this was interphase, that's prophase, that's prophase 1, that's prophase 1, that's prophase 1, and then that's prophase 1. I don't know why they did so many models to show prophase, but they did. Now, down here, does anyone know what phase that is?
Metaphase.
That's metaphase. During metaphase, and actually that's the other thing I'll point out too. Make sure, because we're talking about meiosis and not mitosis, make sure that when I show you one of these stages, you say 1 or 2, right? So this is metaphase 1, okay? And make sure, like for these other ones, you got to say prophase 1. So this is metaphase 1, and you have the pairs of chromosomes lining up together, right? So that's metaphase 1. What's this?
Anaphase 1.
Anaphase 1, okay? Anaphase 1 the pairs of chromosomes are starting to be pulled towards opposite ends. Here represents telophase 1. Okay? You now have essentially two separate cells. The chromosomes are surrounded with a nuclear envelope. So this is telophase 1. Now for whatever reason, this silly model jumps a couple of phases, and it basically, this model doesn't show you prophase 2, it doesn't show you metaphase 2, but it's actually showing you at this part anaphase 2. Notice during anaphase 2, now you actually split the chromosomes apart, and one of the chromatids goes to one end, the other chromatid goes to the other end. Does everyone kind of remember this from AP1? I know it's complicated, I know it's probably giving you nightmares right now, but, and then lastly, this would be like telophase 2, all right? You've got now 4 cells, each of which is surrounded by its own nuclear envelope. Okay. I would review some other pictures of meiosis, just make sure you're comfortable with meiosis.